everyone, I'm Meffre, and I will present you the area survey of a jellyfish bloom dynamic in a small Mediterranean lagoon. Many jellyfish species produce massive blooms. Their distribution is patchy, and their burst and burn nature makes them difficult to monitor. Traditional surveys using nets and boat transects cannot cover the entire bloom magnitude. So how do we quantify the spatiotemporal dynamics of a bloom? Well, we need to take some altitude. Aerial surveys are adapted to jellyfish patchy distribution. Around 10 studies have been used in drones or planes to study jellyfish blooms from the air. These figures present the altitude and flight time used for each species. Apart from our studies, only 3 other studies have used planes to survey jellyfish. Purcell et al. In 2000 to study Aurelia labiata, Graham et al. In 2003 to study Phylorhiza punctata, and Houghton et al. 2006 to study Cyanea capillata, Chrysaora hysocella, and Rhizostoma octopus. Most of the time, planes are used to study big and very visible jellyfish. The species we monitored is Rhizostoma pulmo. This species has many assets for aerial survey. This is one of the biggest jellyfish species of the Mediterranean Sea, and this species swims near the surface. Rhizostoma pulmo does its entire life cycle in Bages-Sigean lagoon. Bages-Sigean lagoon is located in the south of, oh, sorry, you don't have the, yeah, that's better. Okay, I'll start again, okay. Bages-Sigean is located in the south of France. It's a semi-enclosed shallow lagoon with an area of 40 kilometers square. These characteristics makes it a natural mesocosm ideal for the study of jellyfish. 3 cohorts have been identified in the lagoon. To cover the cohorts and capture the spatio-temporal dynamics of the entire bloom, we did 3 different flights. Okay, so let's move on to the image acquisition. We used a small plane equipped with a high-resolution camera. The plane flew at an altitude between 300 and 600 meters. The entire lagoon was covered 3 times, once by each flight. Each flight lasted for 3 hours. Their pathes looks like this. Each flight represents several thousand pictures. So we obtain photos like this one. The resolution is 10,328 per 7,660 pixels, which means that if we zoom in again, and zoom in a little bit, we can differentiate each jellyfish. For the analysis, we estimated that the minimum size for a jellyfish to be properly detected was 10 centimeters. Now let's see how we analyzed the picture. So the first step is to remove all the elements that can be problematic for the jellyfish detection, such as the coast, sorry, you don't hear, the ceiling layer, and the birds. It looks like this once they are detected. And then it looks like this. It creates a binomial mask with the counted and the non-counted area. The second step is the calculation of the area and centroid of the counted area, here in black. Then we detect the jellyfish in this area. For the entire picture, it looks like this. So for each picture, we obtain the total number of jellyfish. We obtain the total number of jellyfish that we can divide by the counted area to obtain the jellyfish density. For each jellyfish, we get the coordinates So, okay. The total length and the area. So let's move on to the results. To this day, we have only analyzed 1 out of 4 images for each flight. They are represented here by the yellow dots on this map. The lagoon area has been divided by a grid. Each case of the grid is 400 per 400 meters. Then we calculate the average density of jellyfish per hectare for each case of the grid. And to be able to interpret the map, we use an interpolation model. So here you can see the average jellyfish density per 100 meters square, so 1 hectare. In yellow, it's a high density. It's 400 individuals per hectare, and in blue is 50 individual or less per hectare. Okay, so these are the maps of the three flights. The bloom seems to start at 3 different spots in the west of the lagoon, then the jellyfish are gathered to the west. We think they can be trapped here by the currents. This creates a huge bloom patch of 4.8 kilometers square, which means that the bloom of this bloom patch of high density covers 12% of the lagoon. During the last flight, we can see that this patch has been divided and moved south. Another interesting thing to notice is that during the first flight, we observe a huge density in the west at the 3 spots. So that might be the polyps locations, because that would be where the bloom begins. It's especially interesting because non-Rhizostoma pulmo polyps has been found in the wild yet. To conclude, aerial monitoring is very complementary to nets or direct observations. The main limits are the limited detection of small individuals, misidentifications of jellyfish. It's a method that is very weather dependent and costly. Nevertheless, this method allowed us to have the entire distribution of the bloom and to capture the bloom in its entire spatio-temporal dimension, which are data we couldn't have otherwise. It's also precise enough for cohorts detection, so the next step of this study will be to detect the cohorts. Thank you for your attention.